Hey everyone, and a huge welcome to all of our Grade 9 biology students at St. Paul American School, Clark. I am so excited to be your host today as we jump right into this explainer. Our exact production title is G9, Biology, Unit 2, Lesson 5, Cellular Reproduction and Cellular Growth. Today, we're going on a bit of a micro to macro detective story. We're going to figure out exactly why the tiny building blocks of your body behave the way they do, how they grow, and how they eventually divide to keep you healthy. Okay. Let's dive into this. We've got three main required concepts to conquer today. First up, cellular growth, specifically why cells have physical size limits. Then, we'll unpack the actual mechanics of mitosis and cytokinesis. And finally, we'll step inside the biological control room to look at cell cycle regulation. By the time we finish, you're going to have a crystal clear picture of how these three vital processes work together.
So, here is our agenda for the explainer. We'll start with cellular growth, run through the math of growth, dive into mitosis and cytokinesis, and then explore cell cycle regulation. After that, we'll do a quick formative check and finish up with a cellular reproduction recap.
All right, let's kick things off with Section 1, cellular growth, and exactly why cells don't just grow endlessly.
Now, this brings up a genuinely fascinating biological mystery. In the macro world, growing larger usually means getting stronger, right? So why don't our individual cells just keep expanding? Why are most cells so incredibly tiny, usually less than 100 micrometers in diameter? I mean, that is literally smaller than the period at the end of a sentence in your textbook. To picture why, think about playing a game of tennis. If you're playing doubles, you and your partner can easily cover a standard-sized court. But what if that court suddenly, tripled in size? You would be completely exhausted and you'd definitely miss most of your shots. The court absolutely has to stay a regulation size for the game to actually work, while a cell is exactly the same way. It has to remain at a strict, limited size so it can function and efficiently transport all its essential materials.
The big takeaway concept here is the surface area to volume ratio. This is the ultimate limiting factor for cell size. Think about it like this. The surface area is the space covered by the cell's outer shell, the plasma membrane. And remember, literally all the nutrients coming in and all the waste products going out have to pass right through that membrane. The volume, on the other hand, is all the stuff packed inside the cell. For the cell to survive, that outer surface area needs to be large enough to support the volume inside.
To really make sense of this, let's look at the numbers. Section 2, the math of growth. We're going to look at a worked analysis model.
Okay, what's really wild about this breakdown is just how incredibly fast efficiency drops off as the cell gets bigger. Imagine a tiny cell shaped like a 1 micrometer cube. Its surface area is 6 square micrometers, and its volume is 1 cubic micrometer. That gives us a super efficient 6-to-1 ratio. Awesome, right? But watch what happens if that cell just doubles in size to a 2 micrometer cube. The surface area goes up to 24, sure, but the volume explodes all the way to 8. Suddenly, our ratio plummets to 3-to-1. So as the cell grows, its volume increases way faster than its surface area. Because of this, standard diffusion becomes incredibly slow. Even the motor proteins that literally drag nutrients along the cell's cytoskeleton railway, well, they become totally inefficient over those larger distances. If it kept growing, the cell would actually starve or choke on its own waste. There's just no way around the math.
Which leads us perfectly into Section 3, mitosis and cytokinesis, the solution to cell growth limits. So our growing cell is facing a massive problem. Once it hits that strict mathematical size limit, it really only has two options: stop growing entirely or divide to survive. And this leads us to the overall process of cellular reproduction, which we call the cell cycle. Now, this cycle happens in three main stages. First up is interphase. Believe it or not, this is where the cell spends the vast majority of its life. We're talking up to 90% of its time. During interphase, the cell is just doing its thing, growing, carrying out normal functions, and crucially, making a copy of its DNA to get ready for division. After interphase wraps up, the cell moves into mitosis, and then finally, cytokinesis.
Let's zoom in on that second step, mitosis. We can break this down into 4 highly energetic substages. A great way to remember them is just thinking of the letters PMAT. First is prophase, where the chromosomes condense and suddenly become visible. Next is metaphase, where all those chromosomes line up perfectly right down the middle of the cell. Then we have anaphase. This is where the cell's spindle apparatus, which is made of these incredibly precise microtubule fibers, physically yanks the chromatids apart to opposite poles of the cell. And finally, telophase, where brand new nuclear membranes start forming around those two separated sets of chromosomes. Now, this brilliantly illustrates a common misconception that we need to clear up right away because people get these two terms confused all the time. Mitosis is strictly the division of the cell's nucleus and its nuclear material, the DNA. That's it. Cytokinesis, on the other hand, is the entirely separate process that happens right at the very end. It's the actual division of the cell's cytoplasm. In animal cells, the outer membrane literally pinches inward, almost like you're tightening a belt, until it pops into two genetically identical, perfectly sized daughter cells.
Moving right along to Section 4, Cell Cycle Regulation, Controlling the Division.
Okay, so now we have two fresh, perfectly sized cells. But how exactly does the body know when to hit start on this whole cycle? Welcome to the cellular control room. The timing and the rate of cell division are absolutely crucial for your health. Your cells don't just divide randomly whenever they feel like it, they follow incredibly strict blueprints. The normal cycle is regulated by specific proteins called cyclins, which bind to enzymes called cyclin-dependent kinases, or CDKs for short. Together, this dynamic duo operates at built-in cell cycle checkpoints. You can think of these checkpoints basically like microscopic traffic lights. They read feedback signals from the cell and will either trigger a green light to push things to the next phase, or they'll flash a yellow or red light to pause the division if conditions aren't just right.
But you might be wondering, what happens when that highly tuned regulatory system breaks down? Well, that brings us to a very precise scientific definition. Cancer is defined as the uncontrolled growth and division of cells. It happens specifically when those traffic lights we just talked about, the checkpoints and the cyclins, fail to regulate the cell cycle properly. So instead of stopping when the area gets too crowded or when a copying mistake happens, these cells just ignore the red lights. They continue to divide rapidly, endlessly, and completely out of control.
Now, normally, before a cell ever reaches that point, the body has a built-in backup plan. It's called apoptosis, which is essentially programmed cell death. If a cell senses unrepairable damage at one of those checkpoints, it will actually trigger its own destruction for the overall good of the organism. Pretty amazing, right? On a completely different note about cell types, we also need to mention stem cells. These are unique, unspecialized cells, both embryonic and adult versions exist, that possess the incredible ability to develop into completely specialized cells as long as they get the right regulatory signals. They're basically blank slates waiting for instructions.
All right, let's head into Section 5, Formative Check. Time for a little testing of your knowledge. We have covered a massive amount of ground today, from the pure math of cell growth to the complex traffic lights regulating division. So, let's do a quick, non-graded check just to make sure you firmly grasped the core mechanics. Here's the question. Which describes the activities of a cell that includes cellular growth and cell division? Take a quick second to lock in your answer in your head. Is there one overarching term that captures the whole shebang, both the growing phase and the dividing phase?
All right, let's see how you did. The answer is the cell cycle. Awesome job if you got that one right. It is absolutely crucial to remember that the cell cycle is the complete, overarching process. It contains interphase for all the growth, and then it's followed up by mitosis and cytokinesis for the actual reproduction.
Which brings us to Section 6, Cellular Reproduction Recap, a final summary of our required concepts.
Let's lock in our top 3 takeaways from today's explainer. Number 1. Strict mathematical limits on the surface area to volume ratio literally demand that cells divide, rather than just growing endlessly. Number 2, mitosis strictly divides the nucleus through prophase, metaphase, anaphase, and telophase, while cytokinesis steps in at the very end to split the cytoplasm. And number 3, this entire beautiful cycle is kept in check by cyclin proteins and built-in checkpoints acting as cellular traffic lights.
Before we wrap up, I want to leave you with one final, kind of provocative thought. We just established that surface area limits strictly dictate efficiency, right? But biology is incredibly clever. Take the lining of your own intestines, for example. Those cells have these special physical adaptations called microvilli. They're these tiny, finger-like projections that massively increase the cell's surface area for absorbing food, but without really increasing the volume at all. It's a total cheat code. So how might other specialized cells in the human body evolve unique physical shapes to push these mathematical boundaries even further? It's a fascinating puzzle, and one I really hope leaves you eager to dig even deeper into biology. Thank you so much for hanging out and focusing in with me on this explainer today. Catch you next time.